So today we are going to concentrate on the physiology component of the muscle tissue, and we are going to focus more on muscle contraction. So previously we had already mentioned how the muscle cell is now, the skeletal muscle cell is now called what? Muscle fiber. Why? Because it's the fusion of what? Cells, the myoblast cells, making it what? Multinucleated. And it gets filled with all kinds of what? Filaments, right? All kinds of filaments, and these filaments are arranged, they are thick and thin filaments, and these thick and thin filaments are arranged into what we call sarcomeres. So this is what? A thick filament, and that is what? Thin filament. And from one Z disc, okay, from one Z disc to the other Z disc represents what? A sarcomere. And so this is showing us a muscle fiber, part of a muscle fiber. And the plasma membrane is now called what? Sarcolemma. And then the cytoplasm, which is the inside, because it's full of these fibers, which are arranged into what we call what? Myofibrils. So myofibrils is sequences of what? Sarcomeres, made up of the thin and what? Thick filaments. Does that make sense? Okay. So we mentioned how the inside is now called sarcoplasm. And the sarcolemma has extensions, as you can see, the extensions going on that forms what? These T-tubules. And the T-tubules surround each what? Myofibril. Okay? And each myofibril also has the smooth endoplasmic reticulum, which is no longer called smooth endoplasmic reticulum. What do they call it? Sarcoplasmic reticulum. And that sarcoplasmic reticulum is filled with what? Calcium, which we mentioned. Does that make sense? And then we have the mitochondria giving the energy. So the importance of the T-tubules, which is the extensions from the plasma membrane, is so that when there is stimulation, remember, cell membranes have what? Receptors, right? Receptors. So that when the receptor gets stimulated, the impulse can what? Travel through the T-tubules and have access to the fibers or the filament of the myofibrils to bring about contraction. Does that make sense? Okay. So today we are going to focus on muscle contraction, and you need to understand this anatomical architecture in order for you to envision what is going on, to visualize what is going on. Okay. So first, let's look at the sarcomere structures, which we've already actually done. This is showing us the myofibril, the M line suspending what? The thick filament, the Z discs that have the thin filament attached. Of course, the myosin also has the thick filament also has attachment to the Z disc. So let's see how this works. Okay, so let's start from here. So this is showing as the T-tubules, the sarcoplasmic reticulum, and all of that, which we call the terminal system. But anyway, the sarcoplasmic reticulum, we are looking at the membrane. This is the sarcoplasm part of it. Do you see the calcium pump, which will keep pumping what? Calcium to be stored in the sarcoplasmic reticulum. And anytime calcium is needed, the calcium will come out from the calcium gate and enter the sarcoplasm. All right? Okay. So first, let's look at how the thick and thin filament looks like. I've already mentioned this to you when we're doing the structure part. But the thin filament is made up of what? Actin, troponin, and tropomyosin. And the actin is made up of G actin and the F actin, and the F strands of the F actin is held together by this protein called nucleolin. Okay, the active site is on the actin, and the specific part of the active site where the myosin binds to, the myosin head binds to, is the G actin. Okay, the active site is on the G actin. So that's the components of the thin filament, which is what? Actin, tropomyosin, and troponin. So let's look at it closely. These are what? Tropomyosin. Okay? While these are what? Troponin. Are you all getting this? Okay? And these are the F actin, and then the G actin are the ones that the tropomyosin is covering. So the tropomyosin is covering what? These are the actin molecules. The tropomyosin is covering what? The G actin ones. Are you with me? And then the F actins are the other ones that it's not being covered. Does that make sense? So if you look at it, it looks something like this. Okay, so you see these, the insides here. Let me just clean it. Okay, just stay that way and don't do anything else I'm not asking it to do. Okay, so if you look closely, you see these parts, okay, are what? The F what? Actin. Do you all see that? What I'm coloring is what? The F actin, which is being held together by what? The nucleolin. Do you all see that? Okay, and then this part is what? The G actin. Does that make sense? Which is being covered, the G actin is what? Being covered by what? The what? The tropomyosin. Do you all see that? Do you all see that? This is what? Tropomyosin. Are you all getting this? And now, keep watching. I just have 3 colors, so I'll use red. Again, now, this is what? Troponin, right? You see some part of the troponin is attached to what? The tropomyosin. Do you all see that? And then this is another part of what? The troponin attaching to what? The actin. And then there's another part of the troponin. So this part of the troponin binds to what? Calcium. This part is attached to what? The actin. And this part is attached to what? Tropomyosin. Okay, so why am I showing you all these details? The G actin part is where the myosin head binds to, and it has the active site. And this whole thing is what? The thin filament, right? All right, and it has the what? Active site. All right, where the what? myosin head is going to bind to. So what are we looking at here? We are looking at what? Thin filament, right? Thin filament. Are you all getting this? This is what? Thick what? Filament. Thick filament in coils. Do you all see that? Are you all getting this? Okay, now listen to this. So what is myosin made up of? We've already established what the thin filament is made up of. What is myosin made up of? What is the thick filament made up of? The thick filament is made up of the core protein called titin and these molecules, protein molecules called myosin. So is the titin, so if this is the titin when at the ends it gets stretched to form these elastic coils that attaches to what? The Z what? Line. Okay? The titin is the core which gets what? Stretch that attaches to what? The Z what? Line. So this is the titin what? Protein which forms the core. And then the myosin molecules have a tail and a head. Okay, have a tail and a head. So the tails wrap around it, and the heads are what? Hanging what? Out. The heads are hanging out and the tails are what? Wrapped around the titin. Capiche? Yeah, so this is the whole myosin molecule, right? This is the tail. Okay, this is the whole myosin molecule. This is what? The tail, and this is what? The head. So the tail wraps around the what? Myosin, and the heads are hanging out in both directions. So again, you have what? This would be what? The thin filament, right? Have your thin filament, and then you have what? No, of course not. What is this? Which part? What is this? It's the titin of what? The thick filament. The thick filament is made up of what? Titin and what? Myosin. So the core, this is the core, which is what? The titin, where the ends stretch and form the elastic thing. And then the myosin molecules what? Wraps around it, where the heads are what? Sticking out. I can't do the whole thing again. Okay? Does that make sense? Okay? So that's how it looks like. So going back, do you see? This is what? The myosin. You all see that? Do you see the heads are those little lines over here? Do you all see that? Okay, and these are what? The thin filaments. Capiche? Easy, right? Okay, so you all understand this? You all see this? Okay, good. So, as I said, the myosin molecule has a tail and what? Free head, all right? They wrap around, bind to other molecules, and then the head is what we see hanging loose. Okay, so how is contraction taking place? I need you to put on your scientific cups. This is what? I'm drawing what? A neuron, okay? These are the dendrites. Okay, of course, this is an apology of a neuron. Seriously. Okay, it looks like an alien. Anyway, so this is one neuron, and this is, let's say, a muscle fiber. Another what? Muscle fiber. Muscle fiber. Another muscle fiber. And this is the terminals of the neuron. Axonal terminals of the neuron. Okay? Are you all picturing this? Okay. So, I'm sitting down minding my business. This is my chair. This is my hand. And then I tell, and this is my biceps muscle. This is my muscle. So I tell myself in my brain, contract your what? Biceps. So what do you think happens? Information in my brain is going to what? Travel to my bicep, and I'm going to what? Contract it. What do I mean by information from my? So what exactly happened here? I stimulated what? A neuron in my brain when I thought to contract my muscle. So this is a neuron. Remember that it's also connected to what? Another neuron, right? So nerves are made up of what? A lot of what? Neurons lined up in a straight line. Okay? So let me draw some couple of neurons here. Just 3 of them. So these neurons are sending information to your muscle. So look at what is happening here. As soon as a membrane gets stimulated, a channel is going to open and allow sodium to enter the cell. When sodium enters the cell, sodium is going to travel down the axons. Okay, that's when we are saying there's a flow of what? Current. And as it travels down the axon, sodium is traveling down the axon. We call that flow of current, and that is what is bringing about what we call impulse or action potential. There is more to this, but when you are doing the nervous system, we will go into details about that. But just so that I can jump ahead and talk to you about muscle contraction, which is today's topic, I'm not going to skip I'm going to skip the details of action potential and how impulse gets generated. I'm not going to talk about it right now. I'll talk about it another time. So this sodium entering, but let me just tell you the basic thing that will help you understand this. So what happened? I stimulated what? A receptor. A channel open and cause what? Sodium to what? Enter the neuron, and sodium is what? Flowing down the axon to the end of the axonal terminal. And that process is what they are calling impulse or what? Action potential. Again, there is more to it, but let's just take that for now. Okay. When the axon gets to the tip of the neuron, do you have your scientific caps on? It will allow calcium to enter the axonal terminal of the neuron. What is it? What is calcium going to do? Do you remember a cell has what? Rough endoplasmic reticulum? And the rough endoplasmic reticulum, the presence of the ribosome can cause it to produce what? Protein materials that can act as what? Chemicals and all of that. And the Golgi body will what? Package it into vesicles. Do you all remember that when we were doing the cell? Okay, good. So the exact same thing has happened in this neuron, where Golgi body has prepared all these chemicals they call neurotransmitters. Anyone heard of neurotransmitters before? Okay. And these neurotransmitters are all at the tip, are being stored at the tip of the axon. So at the end here of the axon is full of what? Vesicles containing what? The neurotransmitters. Are you picturing this? Because a neuron is just what? The membranes are what? Just extended. To draw it properly, this is how a neuron is just membrane. Remember I drew it some time ago? It's just the membrane has been thrown into what? processes. Are you with me? So it's like this, okay? This is the axon. All these things are what? Dendrites. So it's just one membrane thrown into what? Processes. Does that make sense? Are you all getting this? Okay, so it's just one continuous what? Membrane. Does that make sense? Okay, and these areas have, the membranes have what? The proteins on them act as what? Receptors and what? Channels. So that when a receptor gets stimulated, the sodium channel can open and let what? Sodium what? Enter. While at the tip here is full of what? Vesicles containing what? Neurotransmitters. So back to this diagram over here. When the sodium reaches the tip, when sodium reaches the tip of the axon, it will cause calcium channels to open and let calcium what? Enter the what? Tip of the neuron. Does that make sense? And what is calcium going to do? Calcium is going to cause the vesicles to fuse with the membrane and come out so that the neurotransmitter can come out like exocytosis. Do you all remember exocytosis? So the vesicle is going to what? Fuse and then release what? The neurotransmitters. Does that make sense? And then those neurotransmitters, chemicals, will now attach to what? Another receptor on the next neuron, which will cause sodium what? Channels to open to let what? Sodium enter. Sodium will travel down. Calcium will enter, cause what? The neurotransmitters to be released again, and then it will attach to another receptor, and then sodium channels will open again, on and on and on, until it gets to what? The muscle. So this is muscle fiber 1, muscle fiber 2, muscle fiber 3, muscle fiber 4. Are you with me? And so finally it gets to the last neuron that is going to what? Some of the muscle fibers in my biceps. Remember, my biceps is made up of what? A lot of what? Muscle fibers. Are you with me? Are you paying attention to this? This is an important overview. I'm going to say a whole lot on this page. So keep listening. Make sure you're understanding the story. Okay. So when the final neurotransmitter attaches to the receptor of this neuron, the final neuron that is innervating these muscle fibers, sodium channels will what? Open. The sodium will what? Travel down. Are you with me? And it will get to the tip and also cause the release of what? Neurotransmitters. And then what will happen to those neurotransmitters that get released? They will attach to what? Receptors on the membrane of the muscle. What do they call the membrane of the muscle? Sarcolemma, right? So the neurotransmitter will attach to what? The receptors on the membrane of the muscle. And guess what will happen? Sodium channels will also open on what? The membrane of what? The muscle. And sodium will what? Enter the sarcoplasm and travel through where? The sodium will travel along the what? T-tubules. Remember, the T-tubules are extensions of the membrane. Does that make sense? And what will the sodium do when it gets around the myofibrils within the muscle? Remember, the myofibrils are surrounded with what? Sarcoplasmic reticulum. And what is in the sarcoplasmic reticulum? Calcium. So when the sodium gets there, or the action potential reaches there, it will also cause what? Calcium in the sarcoplasmic reticulum to what? Be released. And it's the calcium release that would help bring about the contraction process, which is the next story I'm going to talk about.
It happens all
Every single. Say that again.
It happens like in nanoseconds.
So fast. Okay? Right? So fast. I want to contract my muscle. I did it. And all this is happening. All the stuff that I'm saying is happening. Okay? So fast. You and then you.
With people.
Oh, I was just trying to understand the last part. The neurotransmitter will attach to
The receptors. They always attach to receptors. So the final receptors on the muscle membrane is what? Sarcolemma. The muscle membrane is called what? Sarcolemma. So the receptors on there will attach to a specific, all receptors bind to specific chemicals. So the neurotransmitter will bind to what? The receptor on the muscle, and it will cause sodium channels to what? Open, and sodium will also enter, which is cause a flow, which is what they call action potential or impulse. Again, there's more to it, but I'm not going to go into those details right now about action potential. Then that action potential or the sodium entering, just like it caused calcium to enter the neurons to cause the vesicles to be released, this time when the sodium entered, it also caused what? Calcium to be released from where? The sarcoplasmic reticulum into the what? Sarcoplasm. Does that make sense? Now, the next step is we are going to see what the calcium is doing. How is the calcium helping contraction? That's the next step. But before I get there, when one neuron plus, not when, one neuron plus the muscle fibers that it innervates. In this case, I have how many muscle fibers? 4. They call that motor unit. Okay, so a motor unit is what? Is a neuron and all what? The muscle fibers that it's what? Innervates. Are you with me? One neuron plus all the muscle fibers that it innervates. But remember, my biceps is being innervated by what? Nerves. And nerves are made up of what? Several neurons. Are you with me? So it means that I will have several what? Motor what? Units in my biceps alone. Does that make sense? Okay. So this one neuron may be stimulated and only the 4 what? Muscle fibers will contract. The other neuron in my nerves may not be be stimulated to allow what? Contraction to occur. So in that way, we don't waste energy, because if I'm holding this glass case, spectacle case, do I need all my biceps to contract to hold it compared to a block or something heavier? Does that make sense? So you don't need all your biceps muscles to contract. So that having different motor units is a good thing to allow what we call recruitment, asynchronous contractions, and all of that, which doesn't fatigue all the muscles. Yes.
This may sound dumb, but why is it that you can't flex like opposite muscles at the same time?
So it's not that. It's not that you cannot. You can, but your brain doesn't allow you to, because your brain is coordinating skeletal movement, because your skeletal muscle is attached to what? That's a very intelligent person actually. Your skeletal muscle is attached to what? Your bones, right? What we are looking at. Of course, not all skeletal muscles are attached to for what you are, for based on the question you're asking. It's attached to your bone, and the movement is said that it should allow flexion and what? Extension. And so when you are flexing here, if this should contract, that wouldn't allow the movement that you are looking for. So the brain doesn't allow that. It's automatically when there is really neurons are being released to bring contraction here, it is causing relaxation. But there are certain ailments. One simple one that you all know. You all have taken tetanus injections, right? Tetanus.
Oh, TB.
Yeah, TB. No, TB is tuberculosis.
Oh,
The
tetanus shots, yeah.
tetanus shot. Before taking that, okay. There's this bacteria. You do this in microbiology. There's this bacteria called Clostridium tetani, and it produces this toxin, tetanospasmin toxin, that can cause generalized contraction in all your body. So it causes lockjaw, so generalized what? Contraction in all of your body. So guess what? This is contracting that is contracting. It can break bones, it can get them to care. Patients die because there's cardiac arrest. Your muscles that are breathing, yes, you breathe, you are contracting, but you can't what? Relax them, so you can't take it.
Is it disease like that, isn't there?
Yeah, that's what I'm telling you. And then they curve. That's tetanus. Yeah, that's tetanus. That's why you need to take the shot, so that You don't get
I
that.
thought that was something.
That's that's what it
That's
is.
what I was gonna say.
So you could get tetanus from
From that bacteria,
more things other than like iron and metal and things like that.
because of that bacteria, those rusty ions and metals, especially in your extremities when you get cuts and stuff, that bacteria um, is anaerobic. It doesn't like oxygen, so it lives in dead tissue, dead debris in your foot area. So your foot area has less oxygen. Does that make sense? So it's able to thrive there. And if it should colonize and form parts over there and release the toxin called tetanus toxin into your blood, then it's going to cause the generalized contraction. So it's not the nail per se, it's the bacteria.
No way to counteract that though.
They have to catch it very quickly
Well, I
enough.
mean, like after it happens.
Yeah, that's why you take the shots.
No, I mean like before. After, okay, so let's.
Well, you mean when you're in the episode of it? They
Yeah.
would try to. It's so fast, so you can go. Yeah.
So does that vaccine prevent it from ever happening, or does it?
After every 10 years, because it produces the antibodies that will neutralize the tetanus plasmid, TIG, to bind to and neutralize the toxin. But after every 10 years, you need to take to because that anti-artificial is passive artificial immunization.
I had a patient with lockjaw because he didn't have his tetanus shot. All they did was give him like a muscle relaxer.
Yeah, I was gonna ask that. You can't just
Why not?
relax and just, like,
Because to relax it, because everything is contracted, it has to. Because the whole thing is causing the problem already. So you want the muscle to relax. Because it's too late, it's binding to the receptor. Yeah.
But that's
But you want to make sure that you don't, the muscle, like, we'll talk about it. We have this enzyme, the neurotransmitter that we are talking about here is called acetylcholine. So what happens is that there's an enzyme called acetylcholinesterase which destroys the acetylcholine so that contraction will stop, will cease after it's been done. And so if the patient is not being given the antibody to block the tetanus plasmid, which is acting like acetylcholine throughout the body and causing trouble, contracting everything, all the muscles, then what is going to happen is that you are just trying to treat the symptoms. You are trying to give something to relax the muscle, but the best thing is to have the shot so that you don't even need to get into that state to begin with. Before now, you have to give something to relax the muscle. So the muscle relaxation doesn't mean it's taking away. The muscle relaxant is being used in such a way that to cause the muscle to relax so that you can actually survive. But that doesn't mean that it has killed the bacteria with its toxins. Does that make sense? So it can reoccur again. And you don't want to keep doing that. You want to make sure you get rid of the bacteria plus the toxins it's producing. Okay, good. So you all are good doctors and health people and don't treat symptoms, treat the problem. Okay, so continuing with this story, the next thing we are going to look at is how is the release of calcium helping with the contraction. I need you to remember this again. This is what? Troponin, right? There's a part of the troponin attached to what? Actin. A part attached to what? That calcium binds to. So the calcium that got released attached to what? Troponin. Capiche? And the tropomyosin, that's a tropomyosin covering what? The active site. Do you all remember? The active site of what? The G actin. So listen to this carefully. When calcium, listen to this, and this is the part of the part of what? The part of the troponin binding to what? The tropomyosin. Are you with me? Okay, because the troponin has three parts. Okay, 1, 2, and what? 3. Are you with me? Let me make it 1, 2, and 3. Number 3 is attached to what? The tropomyosin. Do you all see that? So listen to this carefully. This is what happens. When calcium binds to the troponin, it causes the shape of the troponin to change. Okay, a conformational change in the shape of the troponin. And that will pull this part off. And as it's pulling it, what do you think will happen to the tropomyosin? It will be pulled as well. Does that make sense? Because that part of the troponin is attached to what? The tropomyosin. So the calcium binds to what? The troponin, and it will cause the troponin shape to change. And as the shape is changing and raising up, it will cause the part that is attached to the tropomyosin to be displaced. So the tropomyosin will no longer be covering what? The G-actin, like it was covering before. So let me give you an analogy. Think of I like cooking. This is a container, right? This is a lid. This is the lid. Think of the lid as tropomyosin. And the handle, think of it as troponin. And then my hands, these are my hands. Yes, that's calcium. And then, let me use this color. So this space here will be the active site of the G-actin. Capiche? So my hand, which is a calcium, touches what? The troponin, and I raise it up. What will happen to the tropomyosin? It will also what? The lid will also come off. And what will I expose? The G-actin, the active site of what? The G-actin. Does that make sense? Do you all get this? Okay, so that's what the calcium is doing. The aim of the calcium is to bind to troponin so that the active site of what? The G-actin is what? Exposed. Why? So that you see the cross, this is what? The overlap over here. The overlap over here is what? Cross what? Bridges. Thank you. The overlap over here is what? Cross bridges. Are you with me? Cross bridges is the attachment of what? The myosin head onto the what? Thin filament. All right? So, this is what? Myosin head, myosin head, myosin head, myosin head, myosin head, myosin head, myosin head. Attaching what? The thin what? Filament. Are you with me? The specific part of the thin filament is attaching is what? The active site of what? The G-actin. So that's where what? The head is attaching. Okay? So let me go back here and let's look at this. So all this craziness you are looking at, let's see what is happening here. Myosin head, look at my canvas, myosin head. Myosin head, myosin head, right? Thin filament, thin filament. So the side on the thin filament, where the myosin head must attach has been what? Exposed. Are you with me? Why? Because like I said, calcium attached to what? Troponin, right? calcium attached to what? Troponin, which is the lid. We moved it. The lid is what? Troponin, tropomyosin. Remove the tropomyosin and expose what? The active site of what? The G actin. So therefore, now that it's off, the myosin head can bind to what? It. So that's what you are seeing over here. These tiny, tiny lines are what? The myosin heads. These what? Tiny, tiny what? Lines are what? The myosin heads, and they are binding to what? The thin filament. So I need you to watch me right now. I bind to it. They call it what? Cross bridges. And then I pull it. So I'm pulling what? The thin filament towards what? Each other. Are you all getting this? And that is how a muscle what? Contracts. So do you see? There's what? Attachment of the heads onto what? The thin filament. And now it's what? Pulling it towards this side. So do you see now? It's towards this. The H zone is what? Narrowed than it was before. Do you all see that? Do you all get this? Right? You see how simple this is? Okay. So we've talked about the thin filament. We've talked about how the thin filament is what? Exposing the active side of the G actin, and the myosin head is what? Binding to it. Now we are going to talk about what? The thick filament. How does the myosin head attach in the first place? So are you ready? So let's look at one myosin head. ATP is what? The energy molecule of the cell. The myosin head needs to be what? Energized and ready, so that anytime the active side of the thin filament is exposed, the myosin head will quickly what? Bind to it. Does that make sense? And pull it to bring about contraction. Okay. So the process by which you get the myosin head ready to do that, they call it caulking. And this is what happens. Part of the myosin head has an enzyme that is an ATPase enzyme that is able to split the ATP to form ADP and phosphate. That's how energy gets released. Remember I mentioned that to you, okay? So guess what is going to happen here? The myosin head will split the ATP, and now we have what? ADP and what? Phosphate. Okay? Which will what? Release what? Cause the release of what? Energy. And that energy will energize the myosin head. And that is what they call what? Caulking. So it's ready. So the ADP and phosphate is still attached. So as soon as the active site on the thin filament is what? Available because of what? Calcium. This myosin head, which has been energized, will what? Attach and pull the thin filament. Does that make sense? So it's after it's done doing that, okay, and use most of the energy, now the ADP, while it's attached, the ADP and phosphate will what? Fall off. Some books will tell you only ADP is attached, not the phosphate. Either way, it doesn't matter. There's an ADP attached. Okay. So it will fall off. As soon as it falls off, what happens is that ATP is going to come immediately and attach. Okay? And as soon as ATP attaches, its shape will change and it's going to fall off. It can no longer attach to the thin filament. So right now, it was what? Attached to the thin filament and it did a power stroke. And now the ADP and phosphate what? Fell off and ATP was what? Attached. And so as soon as it fell off and ATP came in, it's detached from the thin filament. Does that make sense?
That's after it touches the.
Yeah. So this breaks, it gets energized. Anytime the thin filament is exposed, it attaches whilst it has the ADP and then it does a power stroke. Then the ADP and phosphate is going to what? Fall off and ATP is going to be what? Attached and then it will fall off as soon as the ATP attaches. So that is when the muscle gets what? Relaxed. And when it gets cocked and then, which is meaning it gets energized and then it attaches, that's when what? It contracts.
So
Okay?
when you sleep, there's like a chemical that's released that stops that from happening.
Say that again.
And when you sleep,
Some of your muscles are contracted when you're sleeping.
I know the eyes contract.
A whole lot. So far as this is relaxed, this is contracted. And there's muscle tone that has to occur as well. So yeah.
Do you reuse that myosin head since it
TP.
yeah, it fall off? Like, does it get reused or?
Oh yeah, the myosin head is there. The purpose of the ATP is to give it energy. So it's combined.
So it falls off, there's no energy.
Yeah, when the ADP falls off, because it has finished using the energy, but it can't. The myosin head always has to have either ADP or ATP attached to it. The ADP is attached to it because it was ATP to begin with, and then it broke to form ADP to get energized, and then it attached and used the energy, and then ATP came to also energize it again. So that's what is going on. It's a cycle. Does that make sense? Okay. All right. So we are going to revisit and I'm going to say everything that I've already said. So what I just explained is called what? Sliding what? Filament theory. Okay. So what happens? The thin filament will what? Slide past what? The thick filament. That's what I was telling you. So you have to, so remember, if that is happening, there will be increase in what? Overlapping. Therefore, to make the H band and I band what? Smaller. Remember we talked about the part of the sarcomere. Okay, because there's increased overlapping going on. And therefore the Z lines will what? Get close to each other because of the overlap. After the A band is what? The size of what? The myosin, whether there's overlap or not. So that doesn't change. Remember we talked about that. All right? Okay. So put on your scientific caps. Picture what? A muscle fiber, which is what? A muscle cell full of what? Myo what? Fibrils, right? Myofibrils, myofibrils, myofibrils. And myofibrils are made up of what? Sarcomere, sarcomere, sarcomere. Does that make sense? Okay, so what is happening? When the Z lines are moving close to each other, the myofibril will what? The whole myofibril will what? Shorten. Does that make sense? So that's what brings about so the muscle to contract. Okay, so this is showing us what I just showed you. First, there's what? The attachment of the myosin head to the thin filament is called cross bridge. Cross bridge. All right? The attachment is called cross bridge. So this is showing the power stroke going on and the thin filaments are being what? Pulled close to each other. So now the I band is what? Narrower and the H band is also what? Narrower. Okay, so this is showing us a myofibril, okay, where it's increased what? Overlaps, and therefore the whole sarcomere is what? Shortening because the Z discs are coming close to each other. So this was what it was before and now it's shorter. Yes. So that's how contraction occurs, just starting here. Okay, good. So now, starting from what I've already said, this part that I told you about, where is my drawings? My muscle contracting drawing, where did it go? Okay. So I'm now going to talk about the neuron. This is the neuron innervating what? The muscle fibers. So what we are going to look at is one terminal area and the membrane. Okay, so I'm going to tell you, I told myself I want to what? Contract my biceps, and I did it. How did I do that? Okay, so I've already explained that to you, but we are going to go through it again, so it will be a repetition of what I've already said. So it's between what? Neurons and what? The muscles. So I told myself contract, and remember chemical what got released from one neuron to the other based on what? Sodium moving, which we call the action potential or the impulse, which there's more to it than I'm saying. So what we are going to look at is the one neuron per the muscle fiber that I mean connection. Okay, so this is what one motor neuron, and this is what the end innervating what the muscle fiber, right? So this is what the myofibrils in there. So we are just picking one tiny bitty part of the neuron and how it connects to what? The membrane of the muscle to see. Remember, this neuron was connected to what? Other what? Neurons. Does that make sense? And those neurons were what? Stimulated. They release what? So originally I stimulated the receptor by my thoughts that I want to contract what? My muscle. And then it caused a channel, the sodium channel to open. Sodium entered. Sodium got to the end of the terminal, caused calcium to enter, and the calcium caused exocytosis of what? The vesicles containing what? The neurotransmitters. And those neurotransmitters in this particular case that we are talking about in muscle contraction, skeletal muscle contraction, is called acetylcholine. So this neurotransmitter acetylcholine got released and attached to what? The receptor of what? The next neuron. Are you with me? And that also caused what? Sodium channels to open. Sodium entered till it got to the tip, and also caused calcium to what? Enter the tip. And then the presence of calcium caused what? The release of what? Neurotransmitters. So this story will continue from nerve to nerve, from neuron to neuron to neuron to neuron until it finally gets to what? The muscle. So this is the end. Oops. I hate this thing when it keeps doing that. No, don't do anything. And no again. And stay there like that. Okay. So this is the membrane of what? The neuron. Are you all getting this? So some books will call it the presynaptic. Okay? And then this is what? The membrane of what? The muscle. Are you all getting this? Okay, the sarcolemma, and some books will call it postsynaptic. Okay, so either presynaptic or synaptic terminal, or what? Motor end plate, or what? Postsynaptic, same thing. And then the space is called synaptic cleft. The space is called synaptic cleft. Do you see these tiny, tiny dots? Are what? The vesicles containing the neurotransmitter. Okay, so let's keep listening. So the neuromuscular junction again is what? The synaptic terminal or the presynaptic membrane of what? The neuron containing what? Vesicles that have what? The neurotransmitters. while the motor end plate is what? The membrane of what? The sarcolemma of what? The muscle fiber. Okay? This enzyme, I'll mention it to you later, acetylcholinesterase. And then the space between is what they call what? Synaptic cleft. So let's look at this again. So this is the, a bigger view is showing us what? The presynaptic membrane, okay, or the terminal, and then this is what? Postsynaptic membrane or the motor end plate. These are what? The vesicles containing what? Acetylcholine. Now, do you all get this? Okay. So this is called acetylcholinesterase. That's what destroys acetylcholine in order to stop the contraction. All right. And these are what? The receptors on the muscle membrane. Okay, so let's continue this. So let's look at how the muscle is going to contract to begin with, or how the calcium gets formed, which is what I've already talked about. But again, I want to repeat myself. First, what happens? Electrical impulse, which is called what? Action potential, which is what I've been talking to you about. It has to do with what? Sodium getting there. Okay, will cause the vesicles to fuse with what? The plasma membrane of the neuron, and by exocytosis release what? Acetylcholine. How does it do that? The action potential will first cause calcium to what? Enter. They haven't mentioned it on this slide. Calcium to what? Enter the axonal terminal, and it's this calcium that will cause what? The fusion and then the exocytosis. There's some proteins, V-glutan and all those kind of stuff, but that is not our level of, we're not going to do biochemistry here. Are you all getting this? Okay. So then what happens? The action, the acetylcholine will enter the what? Synaptic cleft and bind to the receptors on what? The motor end plate, which is the postsynaptic cleft or the membrane of the muscle. Are you getting this? And then what will happen? Sodium channels will what? Open, and sodium will what? Enter the cell to bring about another what? Action potential or what? Impulse. Capiche? And what will the action potential or impulse do? It will travel through the T tube. So I'll mention this again later on when I'm showing you the picture. So let's look at the illustration here. This is what? Showing us what? Action potential getting to what? The end of the neuron. And then the action potential, the presence of what? Calcium is causing the vesicles to what? Fuse to bring about exocytosis to cause the release of what? The neurotransmitters. Are you all seeing this? And now the neurotransmitters will bind to what? The receptors, and also cause sodium channels to what? Open to allow sodium to enter what? The muscle. Capiche? And then the excess neurotransmitters, the acetylcholinesterase, will destroy them. Does that make sense? Okay, the acetylcholinesterase will destroy the excess ones, while the ones that are attached will allow what? Sodium to enter to bring about action potential impulse. So do you see that cholinesterase is destroying what? The acetylcholine, so that contraction doesn't continue on and on and on. Are you all getting this? So as soon as sodium enters, what happens? Action potential is what? Being produced. You see? All this is what? Action potential. Are you all getting this? So far sodium is entered. And the action potential is going through the T tubules. Okay? So let me show you. Do you see? This is what the T tubules, the action potential is going through the T tubules. And do you remember each myofibril has sarcoplasmic reticulum wrapped around it? Okay? Containing what? The calcium. So let's look at it. Do you see? This is what? The sarcoplasmic what? Reticulum surrounding what? Each what? Myofibril. So the calcium in this will come out so that it is close to what? The thin filament or the filament of what? The myofibril. Capiche? Okay. Yes, what was your question?
It's not this page where it binds to the troponin, right?
We are still talking about the nerve getting there.
Oh.
Yeah, so yes, when the calcium gets released, yes, So when the calcium gets released into the sarcoplasm, because the sarcoplasm has what? The filaments, the sarcomere structures there. That's when the calcium will bind to what? The troponin, and the whole thing will what? Contraction will occur. So now we've connected the two together. Okay, so let's go back to these slides. Okay, so as you can see over here, action potential will be generated where? In the motor end plate, which will sweep across what? The sarcolemma and the T-tubule. Okay, will sweep down to where? The T-tubules, causing what? Calcium to be released from where? The sarcoplasmic reticulum. Does that make sense? Are you all getting this? Okay, and then the calcium will be released, and then it will bind to what? The troponin, and then tropomyosin will be displaced, and then the myosin will bind. So this is what I was telling you about the myosin. So a resting sarcomere, how does a myosin bind? The myosin head is what? Energized. We call it cocking. Where does the energy come from? ATP. Breakdown of what? ATP. Like I mentioned, part of the myosin has an active enzyme called the ATPase that is able to what? Split the ATP into what? ADP and phosphate to release what? The energy. And this ADP and phosphate will still be what? Attached to what? The myosin head. Yes. Oh, you're understanding it, right?
Yeah.
Good. So I'm repeating what I've just said. So as you can see over here, this is calcium is going to bind to what? The troponin. This is the cocked or energized what? Myosin head. Ready to what? Bind to the thin filament as soon as the active site is what? Available. Okay. So again, calcium arrives, okay, at the zone of overlap. The zone of overlap is where the cross-bridging can occur. The active site gets exposed when what? Calcium binds to what? Troponin. So do you see I'm just repeating myself? Okay? And so this complex will give a conformational change that will expose the active site. The cross bridge is formed, myosin head, which has been energized, will attach.
The changing is when it's
Yeah. So the myosin head, the stored energy within the myosin head causes the power stroke, causes it to move. Okay, it distorts and moves. Then what happens after it does the power stroke, which is what? Pulling the thin filaments together, the ADP and phosphate will be what? Release, fall off. And then what will happen? New ATP will what? Attach, and then the cross bridge will what? Detach. Okay, which is meaning the myosin will remove. And then if the active site is still there because calcium is still binding, then when it gets energized, it will bind again. So the myosin head is always energized. It's just waiting for the site to be exposed and to attach. But when the site is not exposed because there's no calcium, although it's energized, it will still stay there because it can't bind. Does that make sense? So far as there's calcium, that place, if it's exposed, the myosin head will keep being what? Energized and binding.
It's like
And
when you're
contracting.
shivering.
And we will talk about contraction, we'll talk about tension and all those things. That's the next thing I'm going to talk to you about. Okay. So myosin reactivation is when new ATP is what? Broken and then it recoils. Does that make sense? You see? It's very simple. Okay. So the cycle will repeat so long as there's what? Calcium. Okay, so long as there's calcium. So how does the contraction end? The calcium channels that are releasing of the sarcoplasmic reticulum, that are the calcium gates that are allowing calcium to come out, have to be what? Closed first. But when the gates are closed, there's still calcium in the sarcoplasm, right? So remember there's what? Calcium pump, that pumps calcium from the sarcoplasm back into the sarcoplasmic reticulum. That pump will what? Pump the calcium that is in the sarcoplasm back into what? The sarcoplasmic reticulum. Does that make sense? And then the acetylcholine needs to be what? Destroyed or removed by going back into the presynaptic, being reabsorbed. So acetylcholinesterase is what? Destroys the acetylcholine so that it will allow contraction to continue. Because you can destroy the acetylcholine, right? But if there's calcium still present, this will still contract. So the calcium needs to be what? Pumped back inside the sarcoplasmic reticulum. Okay. So,
You know people who have like random muscle spasms.
Or like seizures.
That's just like too much
yeah,
calcium.
it could be calcium. No, calcium cannot be too much because your body will store it in your bone. But it could be leaks of or thoughts, stimulation. You know how like people epileptic seizures, some people specific things trigger
Like,
seizures, light or sharp noise, and so it could be anything. It could be different things. Yeah.
Like what a cramp is just muscle that's just.
Cramps and all those. I'm about to talk about it. Okay. Because they are different. They are different. So that's the next story, and it's pretty interesting. You see how I was trying to let you like put on your scientific caps and imagine and look at the whole myosin thing. It will help you to the cross bridges and all that. I totally want to talk about it now before it's time. Although I know I have 5 minutes, but I may use 10 minutes. But trust me, I want to talk about it now very quickly because you can imagine it all right now. So let's talk about it, right? Okay. And all this will put it to face. But just a quick review of what we said. Neural control, what happens? The action potential occurs, causing the release of the neurotransmitter, which enters the synaptic cleft, binds to the receptors on the motor end plates, causing what? Sodium channel to open to bring another action potential, which will travel through the C tubules and cause the release of what? Calcium, which will then bind to what? Troponin and bring about the sliding process. Now, listen to this. Tension. I want you to, although they are saying tension is tension means pull. That's what it means for sure. But this is a mental picture I need you to get. Think of tension as when the myosin heads attach to the thin filaments without pulling it. Are you with me? Again, think of tension as what? When the myosin heads attached to what? The thin filament without what? Causing the power stroke, pulling the thin filaments together. So just imagine this. When the myosin head attaches to the thin filament, there will be some pull, right? Torque, pull. So that's what they call what? Tension. So you tell me this. If you only have 2 myosin heads attaching to the thin filament compared to 4 myosin heads, which one will have a higher tension? The 4. Perfect. You are ready to understand the rest of the information I'm going to give you. Trust me on that. Okay. So, tension versus muscle length. Okay? Tension versus muscle length. So I'm going to use, you see all this description over here? I'm going to use this diagram to describe it to you. This is perfect diagram. So watch this. This is normal. You know how they tell you stretch your muscle before you exercise? What do they mean by that? Release it so that the overlaps are not too much. Now what do I mean by that? Let's look at this and then you understand what I'm saying. Do you see this number of myosin heads and this is, let's say, normal resting, right? Normal resting. So there is good amount of what? Overlap. Do you see that? There's good amount of overlap. But look at this muscle length. This overlap is not enough. Do you all see that? It's not as much as what? This overlap. Do you all see that? Okay, so it means that here there will be what? More what? Or maximum what? Number of cross bridges compared to what? Here there will be reduced what? Number of what? Cross bridges. Are you with me? So this one will have more tension. That's why you see it's over here. This one will have what? More tension than this one. This one, the tension will be what? Less, because there's less cross bridges that can occur because there's less overlap. Are you all getting this? Perfect. Now look at this one. Oh my gosh. There's almost no what? Overlap. Therefore, if there's no overlap, there will be no what? Tension. There's hardly any cross bridges because it's overstretched. You don't see much overlap. Do you see that? So do you see this is what? Tension. So it's really like the length is really, the tension is really down there because although yes, it's stretched, but there is not enough what? Overlap over there. So you picture this with me. With this particular one here, if let's say there was only one myosin hair that was available, when it attaches to the thin filament, can it pull it effectively? So overstretching, okay, you let me finish before I start talking. Okay, now look at this one too. You need to what? Stretch your muscle, but look at this one. Look at this one. This is what? Z disc, right? This one there is what? Maximum what? Overlap. Do you all see that?
Yeah.
To such an extent that the thin filaments are even what? Overlapping. Are you seeing this? And what is happening here? The myosin head is actually the part that is not called is actually right at the end. So there's no room for even what? Moving anything. Do you all see that? So this one is not the tension. This one, it's just jumped against the Z line, said that there can't be even any pull whatsoever. Does that make sense? So there's an overlap, there's a cross bridges, but even if it attaches, it's just holding it without even pulling. Does that make sense? So the tension is also low because it's just jammed. So when your muscle is jammed like this and you want to contract it, you cannot because there's no more place for anything to go. Does that make sense? So that's why you need to what? Stretch before you exercise, just to bring it to this level. And this one is also decrease in what? Resting sarcomere. So although this one is a little high, but at least you can, although it's also overlapped, but it's not jammed like this one. So yeah, you can produce some what? Tension, but the best one is when it's in its optimal stage. Does that make sense? Okay. So, so having said that, when we say a twitch is what? Contraction, relaxation. Are you with me?
Yeah.
And the duration of a twitch depends on the muscle type and the size and different things. So your eye muscle is small, right? So a twitch is what? Really what? Short. But your calf is full of what? Muscle fibers and hence a lot of motor units.
Like calf twitches.
Right? So it's the tension may be the same, but the duration is what? Longer because it's more muscle and different things. So a twitch is just telling you what? Muscle contraction. And usually a small twitches depend on the number of motor units that are
I
contracted.
just keep pushing down.
Yeah, sometimes it can happen. So basically, they say latent period, contraction period, relaxation period. What does that mean? Latent period is when the action potential is about to bring about the release of what? Calcium. And contraction is when calcium binds and then there's what? The contraction. And relaxation is when calcium falls off and then there's no longer cross bridges. Simple. Do you see how simple this is, which you already understand? Just hold on 1 second. So this is showing us the contraction phase and what? Relaxation phase. Now, this part, the tension thing this is the last thing I'm going to say, then we are off. This is we are looking at two things. Two factors that will determine amount of tension produced in the skeletal muscle is the frequency of stimulating and the total number of fibers that are being stimulated. So I need you to put on your scientific caps here. A twitch is what? Contraction and what? Relaxation. So there is a kind of stimulation that is frequency that is back to back in such a way that the stimulation of the skeletal muscle fiber occurs immediately after relaxing. So I need you to picture this with me. Let's say that you had two cross bridges, okay, and then it contracted. So when it contracted, it pulled the thin filaments together. At least it increased the number of cross bridges as it was pulling it together, right? So let's say it was two and two more joined, but those two more did not attach necessarily. They just joined the whole overlap. Are you with me? Because the more you pull together, the more they overlap. Are you picturing this? So keep picturing. Now, and then as soon as it relaxes, what is relaxation? When there is detaching. Now, I'm not saying a stretch and bounce back, just detaching, right? So as soon as it detaches, you send the stimulation again. So how many more will form cross bridges? Four more. So if four more form cross bridges, then the contraction will be what? Greater. So with trepé, this is what is happening. Immediately after relaxation, when there's a stimulation to bring contraction, it increases. Do you see that? And then immediately it detaches, you do it again, it increases. So it increases like what? Do you see that? It keeps what? Increasing. Are you all getting this? Okay, it keeps increasing till it gets to what? A maximum. Why? Because the total number of cross bridges have already been reached. If there were 10 cross bridges, all of them are now what? Attached. So they pull, and when they detach, you stimulate again. It's the same 10 that will pull because there's no more extra. Does that make sense? So that's when it gets to what? A constant.
Is that involuntary or voluntary?
It's voluntary, but voluntary because you are dealing with what? Skeletal muscle. But voluntary doesn't mean that you can't have leaks of what? Neurotransmitters that you do not control. It can happen that way. Does that make sense? Okay. And then we have wave summation. Again, contraction relaxation, but wave summation is before relaxation. So again, picture two cross bridges. You pull. It doesn't detach. And then you what? Stimulate again. What do you think will happen? The other cross bridges that were formed, the other two that came in when you pull together will now what? Detach. Sorry, will now what? Attach. So now you're having a total of what? 4. And then they will what? Pull together again, and it will recruit what? More overlaps will occur. Does that make sense? And when you stimulate again, then they will also what? Attach and pull. Does that make sense? Are you all getting wave summation? So how does it look like?
Isn't it the same as trepé?
Trepé is they relax, meaning they detach. So trepé looks like this. I attach, I pull, and detach. I detach, but I don't bounce back. I detach, and then the ones that overlap when I pulled will now attach again. Okay? But with wave summation, this is what is happening. I attached, two attached, right? And I contracted. When I pulled together, power stroke, there were 2 more overlaps that came in. Are you with me from each end? But before I what? Detached, I sent a stimulation that caused the other 2 that overlap to attach and contracted again.
So it's like
So it's really
Yes.
wireless.
So it looked like this. Over here, I didn't allow relaxation to occur. I immediately what? As soon as it detached, before it bounced back, I sent what? Stimulation, and it caused what?
Oh, it just keeps building up.
So it's like
So this is how it looks like. Do you see?
in the
then it goes
middle.
back like
In the middle,
Yeah.
yeah, just detach before it bounced back. So you see, before it fully relaxed, I what? Send the stimulation again. Do you see? Does that make sense? So that's wave summation. So with wave summation, we can have incomplete or complete. Okay, incomplete or complete tetany. Incomplete or complete tetany. Incomplete wave summation it looks like this. Do you see? It goes, does wave summation, but now it will get to a time where all the cross bridges would have been formed. So it maintains what? A constant. But with tetany, at least this one, there was what? Some detachment going on, right? But it didn't allow it to fully what? Bounce back. But guess what? Complete tetany is like no room for what? Relaxation whatsoever. So it's the same thing, but it's just contraction continuously. Yeah, back to back to back. No room for that. Okay. So so that's it for now. So that's it for now. And yeah, at least this part I needed you to keep picturing the sarcomere and all of that. So the next time we meet, we'll talk about motor units. Okay.